Hello everyone, welcome back to the channel. For those of you who may be new here, my name's Zach Hancock. I'm an evolutionary biologist who specializes in population genetics and phylogenetics, and am currently a postdoctoral researcher at the University of Michigan. On this channel, you'll find a mix of evolution education, the history and philosophy of science, and some pseudoscience debunking, specifically aimed at young Earth creationist, race realist, and eugenicist. And there's a surprising amount of overlap between those categories. As many of you know, I've been in the field collecting samples for a large-scale population genomic study on coastal crustaceans, and have finally made it back and settled in to actually get back to producing content. If you don't know, this project is part of a larger documentary that I'm putting together to demystify the scientific process, especially how we study evolution for the general public. So the entire experience was recorded, and along the way, I managed to bump into some other popular scientists and YouTubers that a few of you, I'm sure, will recognize. Get hyped, folks. It's going to be awesome once all of this is together. In the meantime, let's return to some of our regularly scheduled programming. A few days ago, a video suggestion came across my screen with the intriguing thumbnail, "Stop Calling DNA Junk." It turns out the video was an ICR interview with Clemson-educated geneticist, a Dr. Jeff Tomkins. I thought I'd take this opportunity to inject myself into the conversation, answering the interviewer's questions as well as responding to and clarifying Jeff's responses. So, buckle up, as I'm sure this interview will be immensely enlightening and not at all strawman the definition of junk DNA.
Hello everyone, welcome to the Creation Podcast, a show where we discuss the science that confirms Scripture. I'm your host, Lauren, and I'm here today with ICR's geneticist and Director of Research, Dr. Jeff Tomkins. It's so good to have you here today, Dr. Tomkins.
I am so happy to be joining the two of you to discuss this incredibly important and often grossly misunderstood topic. Junk DNA is a subject that could really use some clarity, and I'm sure the three of us, including two PhD geneticists, am I right, Jeff, can illuminate the audience on this subject. It would be incredibly irresponsible for us to purposefully mislead the public on the definition of a scientific concept or on the data supporting said concept for purely ideological and religious reasons, as I'm sure Jeffrey knows. And Jeff, I'm quite familiar with your work, and so I know you never purposefully mislead people.
It's great to be here.
Jesus. Was that a smile? Stuff of nightmares.
So today I wanted to talk a little bit about junk DNA, or rather the DNA that many people try to pass off as junk. From my pretty basic knowledge of the topic, only a small portion of any given creature's genome is responsible for coding for proteins. That leaves a lot of DNA left over, and many people try to say that that is completely useless or junk. So where did that idea come from?
Lauren, I need to quickly point out a misconception in your question. So, junk DNA doesn't refer to all non-coding regions of the genome. In fact, the man that popularized the term, Susumu Ono, was a clinical geneticist and well aware of the plethora of regulatory regions and RNA genes in the human genome. He even wrote about these at length in the 1972 article, An Argument for the Genetic Simplicity of of man and other animals, and in his 1972 seminar to the Brookhaven Symposium, in which he argued for widespread junk DNA. So, while it's a common misconception that junk DNA refers to all non-coding parts of the genome, that's just historically inaccurate, as I'm sure Jeff agrees with, right?
Well, we have to go back in history to really the late 1960s to see where that idea came from. So evolutionists began proposing a model called the neutral model of evolution. So this idea basically says that much of the genome is just neutral. It's free to evolve and pop out new genes and new sequences that would, provide something for the mystical agent of natural selection to act upon.
Well, sorry to interrupt you there, but that's a bit misleading. For one, if natural selection is acting on it, it's not neutral, is it? Second, the neutral model of evolution you're referring to is the neutral theory of molecular evolution, which was actually focused on neutral variation, not strictly on functional genomic fractions. In fact, that variation can itself be in functional regions. Most early genetic studies were looking at protein allozymes, which are just different conformations of the same protein. They function exactly the same, but there are non-synonymous substitutions that have slightly changed the structure. Neutral theory posited that this variation has no fitness effect. The entire genome could be functional, and neutral theory could technically still apply if most of the variation between individuals doesn't influence fitness. And why did you refer to natural selection as a mystical agent? It's as if you don't think there are genetic variants that contribute to differences in fitness between individuals, and that those differences cause one variant to increase in frequency at the expense of the other. What's mystical about that?
And so this was before the days of DNA sequencing even. So that's where it really got going, was back then. But then as scientists began to develop DNA sequencing, which was very crude in the beginning, in the 1970s and the 1980s, they began discovering that genes, especially in what we would call eukaryotes or creatures that have a nucleus and have individual chromosomes, they began discovering that genes were in pieces, as they originally said. So you would have a gene, but only certain parts of that would code for protein, and the intervening parts did not. And they originally thought, oh, this is junk, and it fit well with the paradigm of neutral evolution.
If I could elaborate just a bit on Jeff's statement here, he's referring to the observation going back to the 1960s that even for regions that are transcribed and destined to become proteins, only part of the original transcript codes for functional protein. So when the original mRNA transcript is made, it must be processed, in which spliceosomes come and cut out large chunks of what we call introns, and then splices together the remaining sequence, what we call exons. Even of those, only part are actually translated into a protein. Studies on ribosomal genes in the late 60s found that the intervening regions, introns, were almost always destroyed after being spliced out. So, yeah, hard to see that they'd be anything other than junk. Of course, not every intron is destroyed, of course. Some are retained and become things like microRNAs, but that's a pretty small minority, as I'm sure Jeff knows.
And then as DNA sequencing began to progress, they began discovering even more parts of the genome that did not code for protein. So when we get to the Human Genome Project, which really got going in the 1980s and is actually still going on today, they began discovering that less than 2% of the entire human genome actually coded for protein.
Wow.
Well, honestly, we didn't need the Human Genome Project to tell us that. In fact, we knew in the 1960s that only a small fraction of the human genome would be protein coding just based on the human mutation rate. Geneticists like Herman Muller, Moto Kimura, Thomas Jukes, Jack King, and others showed that the number of protein coding genes in humans must be less than 30,000. If it were greater, there would be too great of a fraction of the human genome capable of having deleterious mutations, and the mutation load would be so high that humans just literally couldn't exist. A lot of molecular biologists disagreed and thought that humans are so complex, we must have hundreds of thousands of genes. And there was even bets going into the Human Genome Project about how many genes we'd actually find. In the end, the evolutionary biologists were right. In fact, the number has fallen by almost 10,000 since the initial reports to likely less than 20,000 genes. For context, that's the same number of genes as a nematode worm.
And so that began to fuel the idea of this neutral model of evolution that was begun in the 1960s, and it seemed to validate it.
That would seem very validating at first glance, but is all of that DNA, the 98%, is all of that useless? Is it junk?
When the first draft of the human genome was produced in 2001, scientists said, look, we've got all this great information, all this DNA sequence. We can only identify less than 2% that codes for protein. What is the rest of it doing?
No, Jeff, you're making the same mistake Lauren did. You know that junk DNA does not refer to the 98% of the genome that doesn't code for proteins. You have a PhD in genetics. I assume at some point in your 10 years of schooling, someone explained to you what junk DNA is. It's in your head, Jeff. I know it's in your head. Just pull it out.
And so they started a separate project called the ENCODE project, which it's an acronym, it stands for Encyclopedia of DNA Elements. And the ENCODE project received huge amounts of funding, just like the Human Genome Project did. In fact, it was part of the Human Genome Project that was added on in about roughly 2004. And the first papers that began to be published out of this ENCODE project in 2007 were very shocking. They actually found out that almost the entire human genome was being transcribed or turned into RNA end to end. In fact, one paper called it a RNA machine. The human genome was an RNA machine. And so it looked like the entire human genome was functional.
Whoa there, Jeff. Back up a minute. The ENCODE consortium did indeed find that there was around 80% of the human genome transcribed, but that tells us exactly nothing about how much of it is functional. And maybe we've gone on long enough without addressing the elephant in the room, yeah? Sorry, Lauren, not to tell you how to do your job and all, but we really should have began this with what is biological function instead of talking about coding versus non-coding DNA. Because, junk DNA is about biological function, not about coding versus non-coding. The simplest way to think about biological function is to ask, is this necessary for my survival and reproduction? If a particular stretch of DNA can mutate in any possible way, a deletion, duplication, substitution, whatever, and not affect your health or reproduction, then that stretch of DNA is not functional. Now, that stretch of DNA may do things. DNA is a biomolecule, after all. It's going to undergo biochemical reactions. It's going to fold in certain ways, it's going to associate with histone proteins, it's going to bind various enzymes, etc. But none of that tells you if any of those biochemical reactions are actually necessary for your survival.
Wow, so it actually fills a very, very crucial role instead of just being there as junk.
Oh, exactly. In fact, there was one paper that was published, and they found out that all of this RNA that was being transcribed or copied from DNA, much of it was required for chromosome stability. So these RNA molecules would be transcribed, and they would literally form a matrix around the chromosomes, a functional structural matrix that allowed the genome to function, that allowed it to assume certain RNA or conformational structures.
Again, we're conflating biochemical activity with function. One of the things the ENCODE project did was look at genome conformation, including histone modification and chromatin structure, but they provided no evidence that these modifications were necessary for biological function. In fact, this study from 2012 looked at the impact of various different histone modifications and found that only 142 out of the 8,436 possible combinations, or less than 2%, had a significant impact on gene expression levels. So it's very unlikely that these histone modifications that were studied by ENCODE are actually necessary to maintain function.
And so that's the thing. RNA does so many more things than just code for protein. In fact, one of the interesting things that was discovered in the ENCODE project was that there was over twice as many genes that coded for RNA that was used directly by the cell in some way than there were protein coding genes.
Wow. Okay. So we've talked a little bit about human DNA. Is that the same between creatures? What does that look like as far as non-coding DNA portions across different creatures, across species?
Well, across animals, the human genome functions in a similar way, especially across different mammals like rabbits and creatures that we're more similar to in our biochemistry. But even in plants, we see the same thing. We see genes in pieces, as it were, and we see a lot of regions of the genome that do not code for protein. They code for functional RNA, or they might have regulatory regions around the genome that don't code for protein as well. So these would be like a light switch. So when you walk in a room, you flip the light switch and it turns on the lights. Well, there's a lot of regions in the genome that are basically switches and control regions. In fact, there's millions of them in the human genome that turn off and on things, that tell certain parts of the genome what to do, that turn on genes, that tell the genes how much product or RNA to make, how long to make it, how fast to make it. And so it's very complex what's going on in the genome with all of these regions that do not code for protein, that were once thought to be junk.
Jeff, you seem to be insinuating that the number of regulatory regions of the genome exceeds the number of protein coding regions by a factor of almost 50. Do we really have any evidence that the coding portion needs that much regulation? Lauren's question was about other animals, so let's think about another organism, like, say, the group of crustaceans I work on. They're called hosteriid amphipods, and they live on sandy beaches and have really, really big genomes. One of them, Hostorius canadensis, has a genome that is 14 gigabases, about 4 times the size of ours. If we assume they have around the same number of protein coding genes as us, then the coding portion of their genome is well less than 1%. It also means that if we are to believe Jeff, and I know of no reason we shouldn't believe Jeff, these little crustaceans are far more phenotypically complex than humans because, after all, they need 4 times the amount of regulatory DNA than humans. I'm cool with that, but I suspect the two of you aren't.
Wow, that's amazing. That's amazing. Kind of changing gears just a tiny bit. What about pseudogenes? Can you tell us a little bit about that?
Yeah, pseudogenes were genes that had similarities to protein coding genes. And scientists at first thought that they were junk because they didn't code for protein. Now we know that almost every pseudogene that's been studied is actually a functional RNA coding gene. In fact, many of these pseudogenes produce RNAs that actually regulate the protein coding gene that pseudogene is similar to.
Obfuscation. The action of making something obscure, unclear, or unintelligible.
And so pseudogenes are turning out to be very important for human health. When there's mutations and problems in certain pseudogenes, it can lead to heart disease and diabetes and all sorts of different health problems in humans. So pseudogenes are now actually, most of them that we have studied are actually functional what we call long non-coding RNA genes that produce functional RNAs that do really important stuff in your cell.
You really are a weasel, aren't you, Jeff? Lauren, if I may try and salvage this description, pseudogenes are regions that resemble functional genes, but they are, in one way or another, broken. For example, they lack a promoter, so they can't be transcribed. Or they have a nonsense mutation that causes a premature stop codon, and so the resulting product is truncated. Often, there are multiple stop codons, such that the actual protein, if it were to be transcribed and translated, would be only a few amino acids long, way too small to be a functional product. In fact, remember how before we said that it was thought that there were 30,000 genes in the human genome, but now it's like less than 20,000? This is because we've found that at least 10,000 of the originally annotated genes are pseudogenes. A simple computer program designed to identify genic regions can't tell the difference, and so will zombify genes. That is, it'll resurrect genes that are dead because they resemble functional genes. Apart from the mutations mentioned above, pseudogenes have the telltale signature of a lack of selective constraint. Remember, for a region of the genome to be functional, you need to be able to disrupt that function via mutation. But when we look at pseudogenes, we see a ton of genetic variation between individuals. That is, lots of mutations have accumulated that appear to have no effect. Let me give you a very specific example. Across your genome are what we call numites, spelled N-U-M-T-S, which is short for nuclear mitochondrial elements. These are genes that have been transported from the mitochondria into your nucleus and inserted into your nuclear genome. If you use a computer program that is coded to identify mitochondrial genes from a set of sequences, it will annotate them as mitochondrial genes because of how strongly they resemble them. But when you actually evaluate these sequences, you find that they are littered with stop codons such that they cannot form a functional product. Furthermore, many of them are polymorphic in the population. Some people have them and some people don't. Hard to imagine something that is essential to human health being optional. Man, Jeff?
Okay, so we're finding these elements that we thought were just kind of throwaway are actually completely crucial for various things that our bodies are doing.
Yeah, absolutely.
Wow, that's amazing. So can you talk a little bit about transposable elements and what that looks like?
Well, there's many different types of what's called transposable elements. At first, they were called jumping genes, and so transposable elements are in different categories, and it can get very complex in trying to explain all of the different categories. So I'll try and make this as simple as I can. Most transposable elements are actually not jumping around or doing anything like that.
So transposable meaning it jumps around to a different portion, or what does that?
Right.
Can you define that a little bit?
Yeah, they were first really characterized in corn.
Okay, doesn't it seem like he's really fumbling with this question? I mean, this guy has a PhD in genetics from Clemson, and Lauren thinks she's throwing him a softball, and he's just tripping over himself. Let me help you, Jeff. Transposable elements are parasitic DNA, whose sole function is to increase the number of copies of themselves in your genome. Just like any other parasite, they persist in a host until they kill the host. For example, they overwhelm the host genome and cause literal extinction, or they reach a sort of coexistence equilibrium. They persist with minimal impact as tagalongs, commensals, if you will. And they exist in different categories, as Jeff said. These categories are based on how they undergo transposition, how they jump around. Some are capable of transpositioning all on their own, utilizing an RNA intermediate exactly like viruses do. These are retrotransposons. They even form little capsules around their RNA for protection when they leave the nucleus before being reintegrated. Then there are DNA transposons, which operate under a cut and paste mechanism, in which they splice themselves out of the genome entirely as double-stranded DNA, and then get themselves plopped into the genome at a new location. They often hijack the machinery of retrotransposons to do this, and when they reinsert, they are normally truncated, and so they are what we call dead on arrival, meaning that they're no longer capable of transpositioning again because they are lacking a core gene to do so.
That's where it all got going. But now that we've been doing a lot of DNA sequencing, we've basically uncovered many types of transposable elements, and there's many different categories. As it turns out now, looking at the human genome as an example, most so-called transposable elements are not jumping around. They're actually control features of the genome. They have switches and other important things within them that are doing stuff to keep your genome healthy and functioning.
Lauren, if I may balance out Jeff's nonsense with a bit of reality. So yes, most transposable elements aren't jumping around. The vast majority are actually fragments of complete transposons. They are missing genes, promoters, etc. In fact, almost 50% of your genome is made up of defunct mobile elements. For example, 10% of your genome is composed of one type of transposon called the ALU element, which is classified as short interspersed nuclear element, or SINE. These are actually derived from functional RNA genes that have become parasitic and proliferated immensely in the ancestor of primates. When I taught intro bio, we would actually genotype students for the presence of one specific ALU element as a population genetic exercise in teaching the Hardy-Weinberg principle. You see, ALU elements are almost uniformly functionless. A few have been co-opted, but the vast majority show patterns of neutral evolution. What that means is that their patterns of insertion and deletion match the mutation rate. Some people in the classroom had the insertion and some didn't, and we used the frequency of the insertion to teach how to estimate genotype frequencies and statistically ask whether the observed frequency differed significantly from chance. It was actually a pretty cool lab exercise, and you know what? They never differed from chance. You know why? Because ALU insertions are functionless. Now again, some transposons have been co-opted for function. After all, a transposon has a regulatory apparatus or it wouldn't get itself transcribed, and so even if a mutation disrupts its ability to code for one of its proteins, like reverse transcriptase, it still has its regulatory region just lying around unused. If a de novo gene emerges downstream of the transposon, it can utilize the regulatory apparatus of the defunct transposon without needing to evolve one from scratch. Again, as you might expect, this represents a tiny fraction of transposons. Most have no function and freely mutate without any effect on the host genome.
That's amazing. We are very fearfully and wonderfully made.
No, exactly.
So if you're thinking along the lines of evolutionary theory, then over that long of a period of time, all of that non-coding DNA would be mutating, evolving.
Right.
Do we see that happening?
I'm just going to go ahead and jump in here, Lauren, because we already know Jeffrey is going to say some dumb shit. So yes, we absolutely do. In fact, we find that only 5% of the human genome, when compared to the chimp genome, for example, show signs of selective constraint. What does this mean? Well, mutations occur randomly with respect to their impact on fitness. You don't get good mutations just because you need them. And so when mutations occur in functional regions of the genome, they tend to be deleterious, harming the ability of the individual to leave offspring. So that mutation tends to get removed from the population over successive generations by, as Jeff called it, mystical agent of natural selection. But when mutations occur in non-functional regions, they accumulate just by chance alone. These contrasting patterns emerge as a very strong signature in the genome. Functional regions have less diversity and so are constrained across species, whereas non-functional ones aren't, and so the level of divergence in those regions is actually well approximated by how long ago two organisms share a common ancestor. Okay, but if 5% is conserved across species, what makes humans different from other animals? Well, when we look within humans, there's an additional 4% of the genome that shows signs of conservation. That is, those areas aren't conserved in other species, but they are conserved in humans. That's where we get the 9 to 11% genome functionality number from. It's the sum of the amount of the genome conserved across species and the amount conserved within species. So to answer your question, yes, Lauren, we see very clear evidence that non-coding regions, the around 4% in humans with respect to other animals, and the non-functional regions, the around 90% of the genome, have been evolving. Okay, Jeff, what do you got for us?
No, we really don't, because when we compare, say, different types of mammals and their genomes, a lot of these regulatory elements that are non-coding are very similar between these creatures. And so they're not showing any signs of evolution. It's just common code serving a common purpose, just like a computer programmer would use. And so I actually have had to do computer programming in my research. And when I create a new computer program to do something, I don't create a whole bunch of new code most of the time. I borrow code from other programs.
Right, because it already exists.
Right, it already exists. And so I just put it together into a new system.
Why can you not explain biology without referencing computers? Like, creationists, please stop making analogies to inanimate objects. Genomes really, really aren't like computer code. Like you, Jeff, I do a lot of computer programming, and the similarities are trivial at best and like misleading at worst. Take the transcriptional and translational machinery as an example. Studies have found that the misincorporation of nucleotides during transcription and the subsequent amino acid misincorporation during translation can be as high as 10 to the negative 3 per codon. That means that 1 in every 1,000 amino acids is wrong. If we take 100 amino acids as the average protein length, then 1 in 10 proteins have an amino acid that was not specified by the genome. If I wrote up a computer code and 1 in 10 functions produced an output that I didn't specify, I'd say there was a bug in my code. Why are we willing to say, oh, this code has a bug in it, but God forbid we say the same thing about the genome, despite the vast majority of it behaving exactly like an absolute train wreck of computer code? This is in no way diminishing the genetic code. What makes genomes so fascinating is not their perfection, but that they arose naturally, and that they can function despite not being intelligently designed.
Wow, that's amazing. Thank you for touching on that. So over the last couple decades, humanity has increased a lot in our knowledge on genomics, and also with advances in technology, it's made it a lot easier to study this subject. So what do you think, you've touched a little bit on this already, but what do you think just the top takeaways are? What are the most important things that we've discovered about genomics, especially in pertaining to junk DNA here in the last couple decades?
Well, I think one of the most important things we've discovered is the immense functionality of the genome from one end of it to the other. So there are not regions that are just sitting there neutrally evolving, neutrally mutating. It's just not happening. In fact, many scientists now are realizing this, and it's creating a huge problem for evolution because the entire genome now is appearing to be functional. And areas that were thought to be junk, when mutations develop in those areas, it can lead to serious problems. Say in the human genome, it can lead to cancer, diabetes, heart disease, various ailments like that.
I refer you all to the simple fact that no two humans have the exact same amount of genomic content. In fact, two humans can vary in genome size by nearly 200 million base pairs. What is variable, you may ask? If you were to take a guess as to what those 200 million base pairs were, what kind of genomic region they represent, what do you think they would be? Protein coding? Regulatory regions? Telomeric regions? Or would you guess that that variation was in transposable elements and perhaps endogenous retroviruses? What about you, Jeff? What do you think? If you were to take a guess. Obviously, we know the answer. It's in the repetitive, non-functional parts of the genome. Line elements, long interspersed nuclear elements, explain a significant fraction, followed closely by endogenous retrovirus insertions, which make up as much as 9% of your genome. Lauren, the big takeaways from next-generation sequencing is that approximately 90% of your genome is junk. And by junk, we simply mean is unconstrained, non-functional, unnecessary DNA that does not contribute to your health in terms of survival or reproduction. We should not be ashamed of this. In fact, this is an incredible finding that corroborates theoretical predictions made as early as the 1950s. In any other field of science, these findings would be celebrated as an amazing empirical confirmation. It demonstrates that we really know what we're talking about concerning genomic evolution. We often like to dog people from the past for not knowing everything that we now know, but often it seems like they seem to have known more than what we do. Because people, like Jeffrey over here, have either never bothered to pick up a book or purposefully avoided learning about concepts that make them uncomfortable. We've also learned that genomes vary immensely across the tree of life, with some animals, like pufferfish, having tiny little bitty genomes of only 400 million base pairs, while others, like the unassuming amoeba dubia, having enormous genomes of 670 billion base pairs. That's almost 200 times larger than ours. Understanding junk DNA, Lauren, we can begin to make sense of these dramatic differences. We can ask questions such as, what sort of population genetic environment permitted the accumulation of DNA in one species but not in the other? If we reject junk DNA, as Jeff and those like him seem to do, and I lump him in with pan-adaptationist molecular biologists who are a little better than creationists on this point, then we are left with the puzzling paradox that many protozoans have bigger genomes than we do. And that leads to my final point, Lauren. The evolutionary biologist T. Ryan Gregory posed this question in response to the ENCODE project's initial claim of 80% genome functionality, which the popular press often forgot to update to the mere 20% that the lead authors eventually settled on. He called the question the onion test, and it goes like this. The onion test is a simple reality check for anyone who thinks they have come up with a universal function for junk DNA. Whatever your proposed function, ask yourself this question. Can I explain why an onion needs about 5 times more non-coding DNA for this function than a human? You can replace onion with any organism you like that has an immensely larger genome than us. If your explanation can't also explain these phenomena, then it's probably not a very good explanation at all. With that, I think we've heard enough from Jeffrey Tomkins. Junk DNA is one of those topics that is incredibly misunderstood, not just by creationist hacks like Tomkins, but even by science journalists and some scientists who should really know better. I hope you come away from this with at least a somewhat better understanding of what junk DNA is. Thanks so much for being here, and I'll catch you next time.